and we can ease into the day by having you give me some kind of layperson explanation of what memory is. Does anyone have anything to toss in to get us started? How would you describe memory? Make sure I can see the chat.
A collection of all kinds of information in our brain.
Anyone have any ideas?
Can you hear me?
I can appreciate getting the day started with this tough one. Yeah, go ahead.
Yeah, I talked. Couldn't you hear me?
I don't know why I can't hear you.
Oh, no one can hear me.
Yeah, I can hear you.
Oh, you can. Okay.
Let me see.
Uh,
That should be right.
Maybe it's in the
Should be
system because I don't know. So if this one is not working,
Strange.
maybe select same system because then with your computer.
Yeah. So, where is it?
The real tech is yours.
The Realtek is my array here, yeah.
Okay.
And then this is my microphone, so that should work. Strange. Can you test it and see if you can hear this?
So I actually was just hearing from the microphone.
Mariam, can you try speaking one more time?
Yes. No.
Yeah.
Test. Whoop.
Yes. Can you not hear me?
Yep, I can hear you.
Yeah.
I don't know
Great.
why we can't on my computer. I'm gonna see if I can fix that.
Okay.
But go ahead with your
Yeah,
answer.
I said collection of all kinds of information, which can be episodic, procedural.
Collection of information. That's interesting.
Yes, of all kinds.
Very, very objective and rather detached. So collection of information.
I have it. I figured it out now.
You figured it out?
Yeah, it was just
I'm going
down.
to stop my sound then because it might be a bit.
Exactly. Okay.
Now your voices have a little bit echo.
Yeah.
No.
Yeah.
You know, okay.
Okay. Yeah. I think if you, yours now. Yes. Okay. Now let me try your. Say something again, Mariam, just to test.
I am
Yes,
saying something
test.
now, just a test.
Okay. Good. Okay. Cool. Okay. So yeah. So a collection of different information, and then you were giving some more definitional words after
Yeah,
that.
I said different types. For instance, it can be episodic and procedural. Yeah.
Do you have any? How would you explain that without using those kind of keywords?
Those words, for instance, how to play a certain piece on a piano, that's also some kind of type of information. And the
Okay.
information that you have about from the lecture or what you learned yesterday, that's also a certain type of information. And what happened last week, that's also some type of information.
Okay. So a collection of information, and it could be a wide array of kind of concepts of different information. But then, can anyone give me, how would you break down the components of memory? So if you were going through the steps of a memory, what would that look like? Anyone have any ideas?
Perception, then saving it, then into storage, and then the possibility to retrieve.
Nice. Yeah. So I'm going to put this up on the screen now. So yeah. So I think you guys are both very correct. This is there's a vast array of information that your brain can hold, and memory is the process of acquiring that, storing that, and then being able to retrieve it. Exactly as you said, Julian. So encoding, storage, and retrieval are the terms that I've chosen here, but perception is of course necessary for that. You need to first perceive the actual experience that you're trying to remember, and then you encode that into the neural language and store it and retrieve it for later. Okay, so, brought that up too quickly. Okay, so then if those are your steps for memory, then can anyone tell me about learning? How would you describe the process of learning in relation to memory? Any ideas? Let's see, who have I not heard from very much?
Learning results, some type of memories.
Yes. So that can be the process of, yeah, let me think about. Yeah, memory can be, of course, the result of learning. But how would you describe what the experience of learning is, or maybe even the mechanism of learning to someone? Yeah,
Maybe.
acquiring new information would be part of it as well. Any other ideas?
Learning would be equivalent to the encoding.
It could be the encoding, but it also could be, I would say, a modification of something. So it's possible that you've already encoded something, stored it, and retrieved it, and you actually just need to improve upon that information in order to get even better at something. So if you imagine your piano playing example, there can be a process of constantly updating that memory. So there's also that component to this. So you can enhance the performance of a particular task, and that will alter the memory, but it can also be the source of a brand new memory. So, okay, so these are some kind of basics to memory, and we're now going to get more into the actual biological mechanism of how memory is formed and how it can be retrieved. And we're going to start by talking about these different types of memory, which Mariam mentioned before. There's actually many different types. You can first break them down into two forms of long-term memory, either implicit or explicit. And so I believe the examples that Mariam gave were episodic, which is actually a memory of events. You can also have semantic memories, which are just pure fact information. It has no relationship to an actual event that you experienced necessarily. You may have it linked in your memory that you have some fact associated with an event. That's not necessarily the case, but you could have some fact like who is the king of the Netherlands. There's just some bit of semantic information that you remember, has no other relationship to an event necessarily, except that when I think of that, I also remember a time when he came to visit Erasmus. So I have those memories linked, but those are two different types of memories. And then you can have implicit memories. And the difference being between explicit and implicit is that you're consciously recalling and you're actually aware of it in the explicit case, and the implicit case, you actually don't need to consciously think about it. And it actually is stored in ways that they're not reflexive, but they are fairly autonomous, which is nice. So once you get really good at playing the piano, to follow that example, you no longer have to think about every single component of your fingers. You don't have to think about what the treble clef looks like or the bass clef. These are things that now just kind of get built into your processing, and that might be a procedural skill, which is controlled by a part of the brain called the striatum, or you may have some sort of associative learning that might have an emotional response or some sort of muscular response, which would be controlled by the amygdala and the cerebellum. Priming is an example that I won't get into in much detail, but priming is basically it's like an expectation setting. So a classic example is if I suddenly jump out at you, then that might scare you. But if I give you a little warning first and then do the exact same thing, you won't be scared by this anymore. And that actually requires a memory immediately, but also a long-term memory. You can prime something for the distant future. I will show you one example at the very end, and that's it. Or actually, it might even be on a different day. Maybe this is a a cliffhanger. I can't remember, ironically. But anyway, so today we will talk about a variety of these different types of memory, and we'll talk about the actual circuit mechanism for them. So if we want to start now breaking down the biology and the circuitry, we need to first be at the very kind of macro level, which is just that you actually have different brain structures that are involved in these different types of memory. So that priming memory is associated with the neocortex. striatum is part of the basal ganglia, which is associated primarily with procedural learning. The amygdala has a lot to do with emotional response. The cerebellum has to do with classical conditioning or associative learning. And reflex pathways are within your spinal cord, and to think of them as a memory is really quite interesting, but it's important to consider that, because really a memory is just a bit of information that is stored within a neural circuit and can be retrieved when it's necessary. So when the doctor knocks your knee with the little hammer, you wouldn't think of that as a memory that your leg moves, but to some extent that's stored information within your neural circuit that can give a reliable response. So these reflex pathways are to some extent an implicit memory. And then you've got your medial temporal lobe, which contains your hippocampus, and that's very important for explicit memories, the semantic and episodic, and we'll go into quite a lot of detail about that as we go. So first, in order to start thinking about and breaking down what memory and learning actually is in a very mechanistic way, we're going to go to a simple example of learning and memory. This is Eric Kandel. He's a Nobel Prize winner. He's also the writer of The Principles of Neuroscience, which presumably you are at least familiar with. And this is him holding a beautiful animal called the aplysia. And it's interesting that he chose this as his model organism, but this is the California sea slug, I believe. Yeah. You should look up what they look like in higher resolution. I couldn't find a higher resolution picture of Kandel holding the aplysia, but the aplysia is quite a fascinating creature. But this was his model organism. So before we get into his actual experiments, I want to ask another question, which is, does anyone have a good understanding of Pavlovian or classical conditioning that they can share? Yeah. Feel free, anyone, to unmute and give an example of that. Let's see, should I call in somebody? Who is that?
The famous example of Pavlovian dogs.
Yeah, so can you go into more detail on that?
Who associated the bell ring with the food. So every time they would be exposed to be fed, first they would hear the bell ring or some kind of other external stimulus, but I think it was the bell. Also can be the flashlight or other perceptual modality. And then from after some time, they were already salivating for the bell, because that would predict, or that would be associated with the food.
Mhm.
So
it's interesting that you said that it could be a bell or it could be some other thing. So how would you term that group of the bell or some other stimulus? What would you call that?
Neutral stimulus.
Yeah, exactly. So some stimulus that would not typically lead to salivation, which to some extent is a reflexive implicit memory to begin salivating when you have food in your mouth. And the fact that it is an implicit memory means that it can be tapped into, and you can actually get that neural circuitry to be activated by other things. So it is, yes, very much an example of being able to learn a new association between something. And so the classical example that you see is often a little oversimplified, but this is roughly true, that this is what Ivan Pavlov found, which is that he was testing and measuring saliva production, which is quite a fascinating original question. Now that to imagine what he's actually contributed to science, he was just measuring how much saliva dogs produce. But he ended up noticing that when the research technician came into the lab, the dogs began salivating well before they were actually given food. And so then he started testing that more explicitly and found that you could associate a neutral signal or a conditioned stimulus with a unconditioned stimulus. And so when we say unconditioned, that is something that is tapping into a reflexive response such that food always produces saliva, and you don't necessarily have to learn that. That's something inbuilt. But a bell does not produce saliva. There's no reason that a bell would cause a dog to salivate, but you can actually link this neutral stimulus to this salient stimulus, and we can call that a conditioned stimuli and an unconditioned stimuli. The salivation to food would be an unconditioned response, so there's no conditioning required. The salivation to the bell is our conditioned response. So even though it's the same response, the fact that it's now happening in response to a previously neutral stimulus means that it's now a conditioned response. So if you need to imagine, how would you design a circuit that would allow for such a thing to even happen? So it doesn't make a whole lot of sense to ever salivate to a bell, except to get your body prepared for something that you really are confident will happen. So there's kind of an expectation that can be set up there. If you imagine that you're looking forward to a reward, if you imagine a series of events, if you imagine that you need to associate some kind of random noise with something important. So the example I always think of there is, imagine when you were very, very young and your parents started saying your name. In the beginning, that made very little sense to you. It was just some random series of noises. But over time, you began to associate that random series of noises with something meaningful actually for you in some way that your parents would associate with you. Learning your own name is not necessarily a classical conditioning bit of information, but it's certainly taking something that was some neutral random sound and associating it with some deeper meaning through learning. So there's many ways that you can begin to associate things that previously didn't seem to have importance with something that is important. And the neural circuitry requires that there be a pathway in place in order for the response to exist and some way to alter that pathway. Okay? So this brings us back to the aplysia, which is Eric Kandel's model organism. In the aplysia, this sea slug has a few different anatomical components that you need to be aware of. It has the siphon, which is part of a release valve for any water that the slug breathes. It has this respiratory gill, and it has this mantle shelf, which kind of holds the siphon. So what they found was, in the beginning, that if they took just a little piece of metal or a piece of plastic and just poked that siphon, that the aplysia would retract everything. It would respond to it by having something of an alarmed response, right? So if you go in and you poke this siphon, you create a gill withdrawal reflex circuit where the gill is shrunken in and pulled back, right? So how would that be possible? That means that there has to be some connection between the poking of the siphon and ultimately the movement of that gill. So if you now think back to the C. elegans example, you can have a sensory neuron connected to the siphon, which is then connected to interneurons and ultimately feeds into a motor neuron that targets the gill, right? Now, one of the early experiments that the Kandel lab did with the aplysia was to test something called habituation. Where they found that if you poke the siphon sufficiently over time, the animal would eventually not withdraw their gill anymore. So the animal has now learned that this tactile stimulus of being poked on the siphon is actually not that important. And so they don't go through the work of withdrawing their gill anymore. And the way you could imagine that happening is that there's actually a variety of ways that it could happen that I hope you can appreciate. But the way that it does happen is that over time, there's a reduction in the release of neurotransmitters onto the next neuron. So the sensation that you get from the sensory neuron, or the sensation that the sensory neuron experiences from the siphon being poked, is basically the same over time. So this is on multiple times of the siphon being poked. This is the action potential waveform that you get at the sensory neuron. And you see that basically that is unchanged. So that sensation is always detected, but it's no longer translated down to the motor response of the gill anymore, because that sensory neuron begins to downregulate how much neurotransmitter it's releasing. So if it releases less neurotransmitter onto the interneuron and the motor neuron, then there will be less activation of the motor neuron in order for the gill withdrawal to be smaller. So in the beginning, you have this large gill withdrawal. Towards the middle of this experience, they begin to reduce how much the gill is withdrawn. And after a while, it's much lower. So this is the EPSP. This is the excitatory postsynaptic potential of this motor neuron, which is just a measurement of how much signal has moved from this neuron to this neuron. And you can see that reduce, and that reduces in perfect time with the gill withdrawal being reduced as well. If you then go back and wait for a little while and let the animal just rest for about an hour and then poke the siphon one more time, you see an increase again in the response here, and you see a big increase in the gill withdrawal. So this is definitely a reflex that will be maintained, and it can be habituated, but it will recover as well. It won't quite recover to the same amount, but it will definitely recover. So this is a simple circuit that could now be tapped into in order to understand what is the actual circuitry involved in a memory. So just this experience is already a learning experience of the realizing that this stimulus is not very important. And so now what we have is this nice pathway from the siphon to the gill in order to test some memory components. We're going to complicate it one step further to get us back to the Pavlovian or classical conditioning model by adding in a stimulus to the tail. Okay, so we still have our siphon, sensory neuron, interneurons, a motor neuron, and our gill. That's still our primary readout, but we also have a tail signal that has its own sensory neuron, its own facilitating interneurons, which also project into this same neural pathway, which means that we have the means to hack into a neural pathway through a different route by poking the siphon again and by stimulating the tail. And what you can find there is that initially you can actually increase the response of the gill withdrawal by combining a stimulation to the tail and a tactile stimulation. And I'll show you a little more detail about that soon. But basically, you can initially find this typical response and then a more sensitized response by pairing these two stimuli, where the gill withdrawal actually increases more, and you can actually increase the sensitivity of the sensory neuron that is detecting information from the siphon. Okay, so how would this actually happen? Basically, if we really zoom in on this pathway, so I'm going to take you back to remember what this looks like. Right here, you have the sensory neuron axon coming down, and you have the axon terminal, and you have a synapse onto the motor neuron, and then you have another axon terminal actually coming down and contacting the axon terminal of the sensory neuron. So you have a circuit in which you have a direct pathway, and you have this little modifying synapse. So if we zoom in on that, this is what it looks like. This is the axon terminal of the sensory neuron. These are all the vesicles with their neurotransmitter. It's actually glutamate. And then on the motor neuron, you have glutamate receptors, which can be activated upon release of glutamate by the sensory neuron. And then at the same time, we have our facilitating interneuron, which is mediated by activity of the tail, signaling onto the axon terminal of the sensory neuron. And this is releasing serotonin, which can activate serotonin receptors. That's what this 5-HT is. That's the abbreviation for serotonin. And then the 5-HT can actually, through a process of an intracellular cascade, can actually increase the amount of reserve pool for vesicle release and increase the sensitivity, ultimately, of the neuron by altering the intracellular makeup. So if this is activated a lot at the same time as the terminal is activated by the siphon sensation, then you can actually increase the amount of glutamate that is ultimately released onto the motor neuron. So let me show you what that looks like in principle. We have our unpaired pathway, which is just the direct pathway from the siphon to the gill, where you have the siphon, sensory neuron, motor neuron. And we've even removed the interneuron from this diagram because ultimately that's not necessary for the principle, but just to remember that it's even more complicated than this. And in this case, what you do is you stimulate the siphon and you stimulate the tail, but you keep them quite distant from each other. So they're not actually overlapping very much in time. So in this case, you have this before training and after training, but you're not actually pairing the two stimuli during the training. So there's no ultimate difference. You have a response from the sensory neuron, and you have a response from the gill motor neuron, and obviously a withdrawal from the gill, but there's no real change over time. However, now, if you pair these two and you stimulate them in a way that's overlapping in time, you can actually enhance the signal, the response from the of the gill withdrawal. So here you have the stimulation of the siphon. This is your conditioned stimulus. Your tail would be your unconditioned stimulus in this case. And then you have ultimately an increase after training of both the sensation of the from the sensory neuron and the size of the gill motor neuron withdrawal. So this is essentially the same model of classical conditioning that you would have in Pavlov's dog, or that you might have in any number of events in your own life. So an example that I gave recently was imagining that you're standing on the tram and you're holding a pole, and suddenly you hear a screech of the tracks, or maybe you see the compartment in front of you suddenly jerk around. You might actually brace yourself without even thinking about it consciously, because you've learned that that means that your own compartment might shake and you want to maintain your balance and posture. So you have also experienced classical conditioning training, whether or not it's your parents saying your name and pairing that with something, or telling you the name of some food that they're teaching you, or just learning to maintain your balance. This is the same sort of circuitry that you see in the aplysia. So, okay, so this combination in time is very important. And so you can imagine that it makes sense conceptually that things need to be paired in time for you to associate them. But the reason that it is necessary is because of the physical molecules that are moving around inside of the neurons in order to make sure that the memory is strengthened. So if, for instance, you have an action potential moving down the axon of the sensory neuron, is now coming to release activity or neurotransmitter onto the motor neuron, you need for all of the proteins and activity within that axon to still be taking place while this neuron is exciting the same axon terminal, in order to increase the amount of protein production that's within the axon. So in this case, what you see is an increase in protein production at the nucleus, which then sends new proteins down in order to grow a larger axon terminal, for instance, or to just add entirely new synapses. And this will actually strengthen in a more permanent way the connection between two neurons. But you need them to be paired in time in order for that to actually happen. So if we go back to this sort of example, here you have always paired shocks. So you have the CS always paired with a US. You may have times when the CS is presented without the US, that's okay, but you always have the US following a CS, and in that case, the strength of the conditioning would be quite high. However, if you have some US presentation not paired to a CS, then your strength of conditioning will be low. And if they're just kind of randomly interspersed, then there will be no pairing, which makes perfect sense conceptually. But physically, the reason for that is that you have actual proteins that are moving through your neurons that need to be interacting in time in order for something to be strengthened. So this addition of new synapses is part of that. You can see that in a variety of examples. So for instance, here you have a neuron that is targeting another neuron involved in some behavioral process. And once a training has taken place where you have a control example and then you can habituate that behavior or sensitize that behavior. What you see is the control example might, for instance, have 2 synapses. A long-term habituation will teach this circuit that it's not a very important connection, and it will reduce down to 1 synapse. A long-term sensitization will convince this circuit that this is a very important signal and will actually increase the number of synapses. In order for that to take place, they need to be paired in time to allow for interaction of proteins and for the actual buildup of new material in order to strengthen the synapses. And those connections can be quite specific. So this is a really basic example of a sensory neuron targeting 2 motor neurons. Here you see the within the in vitro setup here. And what you can actually do is facilitate just one of the synapses, where you actually can you can only increase the activity of one side of it, even though it's coming from the same neuron. And you can leave the other one at the same level. So the way you would do that is with a short-term facilitation by targeting this one synapse with serotonin. You can actually do a long-term facilitation where you target it with quite a lot of serotonin, 5x serotonin, and you can find that you will actually induce a response in a synapse-specific way. You might have some sort of leak as like cross-synaptic facilitation if you pair them properly, but you can also be very synapse-specific in your facilitation. And this means that you can actually mediate learning within a circuit, even though one neuron is controlling ultimately the output of multiple neurons. You can mediate learning by targeting only that one connection. So this is a result of this sort of process of information coming into a singular axon terminal and producing more protein in order to build up the strength of that protein, or of that synapse. So really the way that learning can take place is for a reliable response within a neuron to lead to a larger or smaller response in the next neuron. And this is the basic mechanics of how that would come to be. Serotonin, by the way, is not the only means for that. It's just the example that keeps coming up in this lecture so far. But serotonin is not the only neurotransmitter necessary for that. Okay, so this is the basic biology of how a memory could be formed. We'll get into some other examples of that soon, but by strengthening a neural connection, you are creating a proper learning and memory process. Okay, so now we're going to get into different types and how we know there are different types. So we need to now kind of test what I'm telling you and make sure that we have good evidence for these different types of memories. And the way that we can do that is by studying these different parts of the brain that I'm claiming are responsible. So if I tell you, for instance, that the hippocampus and the medial temporal lobe are responsible for explicit memories, and I tell you that the neocortex is responsible for priming, or the striatum is responsible for procedural learning, the question I would have for you is, how do we know that that is the case? Can anyone imagine different studies that might have taken place in order for me to confidently tell you that? Any ideas? I'm going to bother, hold on, Mariam, I'm going to bother someone I've not heard from yet. I'm going to bother Felix. Do you have any ideas?
Maybe by like trying to teach them the different tasks and then like trying to measure brain activity.
Okay, so
Like,
so actually to teach with potentially in an animal study, it could be done in a human study, to actually teach them a specific type of memory and then record activity and see what part of the brain is working in that case.
yeah, and what you could do is, I think with patients, when part of the brain doesn't work, and then try to teach them different things, and then from there try to see if they, like for example, the hippocampus doesn't work, what type of memories can they build, and what memories can they still build?
Great. Yes, exactly. So that last thing you said is exactly what we're going to get into next with some famous patients in neuroscience. So this is one that you probably already know, who's HM. We can reveal his name now, Henry Molaison, because he's now passed away. And I think the family was fine with him being public. If you already know this, then that's okay. I'm going to go very quickly through this. But essentially, he had a very severe epilepsy. It was a temporal lobe epilepsy. They identified that that was the part of the brain that was inducing the epilepsy and decided to remove it. But in removing the temporal lobe, because it was such a severe epilepsy, they removed quite a lot of tissue. They also removed the hippocampus. They removed other things too. It's not a pure hippocampus removal, by the way, but they did remove the hippocampus. And he ended up with some interesting behavioral results that lead us to, that led in the beginning for the concept of specific memory storage. So this is a nice article. It's actually his obituary in the New York Times. And you can go back and read this to see what people think of him. But basically, he could only remember things that were in the very distant past prior to his surgery, except for other types of memory like implicit memories. So an example of that, so let me actually finish this before I move on. So he had explicit memories of the time before he had this surgery and this removal of his hippocampus, and very detailed things as they're indicating here. He could tell you about the stock market, World War II, and life in the 1940s, but he couldn't tell you if he had met you the day before. So he had several researchers that would work with him that would come and meet him on one day and then come back the next day, and he would have absolutely no memory of that person. But he could learn certain things. So this is the early days of starting to separate out different types of memory. This is an example of the type of memory that he was actually capable of doing. So this is a task where you actually have your hand blocked and you're looking through a spiegel or a mirror, and you have to trace something that's on the sheet in front of you. That's quite difficult, actually. If you imagine doing that, you can't see your hand anymore, and you're looking at a mirror image of your hand, and you have to now control your hand well enough to trace it. And what you can do is you can track the number of errors over time, and you can watch that they actually reduce with training. And this was something that HM was capable of, even though he had no explicit memory of ever having done this task before. When he did it when he went from day to day and actually did this task, he had no memory of it, and he would always tell people this was the first time he was doing it. But he actually showed a learning improvement over time, suggesting that there was a different part of the brain that was responsible for him being able to perform that task. And in this case, this would be something of a procedural learning or a skeletal muscle learning, which would be basal ganglia or cerebellum. And those systems were intact in his brain and allowed him to maintain this type of memory. Okay, so lessons from HM, just throwing this slide in here. The hippocampus is important for obtaining new declarative memories, these explicit memories. And we know that because he could no longer do it. So as Felix said, when patients have a specific dysfunction of the brain, you can look at the specific behavioral outcome. But it's not necessary for non-declarative memories, such as procedural learning. It's not necessary for immediate memories, because he could interact with someone and he could even remember a series of numbers in the immediate presence, but he could not remember it the next day. And so it's also not involved in the storage of long-term memories, because he had these very distant explicit memories from the past. So this one patient gave a lot of information early on that allowed people to begin testing these things in other ways that we can get into quite soon. Another patient example is patient KF, who was in a motorcycle accident and hit his head. And in this case, he had a damage to short-term memory, but he had a relatively normal long-term memory. So this suggested that you actually had the ability to separate out short-term memory and long-term memory, which is a little confusing. But the processes for short-term and long-term memory are actually different enough that they don't necessarily require each other. So you could have a long-term memory of something that you didn't remember very well in the moment. So that was what KF taught us. And one last thing before we take our first break is that you can study memory in animals, and you can even study declarative memories, which is a little confusing to imagine because an animal can't declare anything to you. But with the right experimental design, an animal could declare something to you. And this is the case of that. So basically, you have the animal sitting here, and you present them with two little holes in the ground that you see here. And one of them has an object above the hole, such as the key. And when the animal moves the key, they find a bit of food beneath the hole, okay? And then after some time, you test the animal. You present the animal with the same object, the key, and another object, but now you've put the food under the other object. So over time, the animal will learn that when given a key, the food is always below the other object of a key. So the fact that the animal reliably learns to pick up the other object, even if you change it to any number of things, is the monkey declaring that it remembers the key by not choosing the key. So this is a very straightforward, it's an interesting thing. I'm not sure if, I'm sure it took quite some time to imagine this, this experimental design, but this is an example of how an animal that can't speak can actually declare something to you. Okay, so we can take our first break now and start getting into the anatomy of the hippocampus and the actual circuitry underlying how memories could be initially formed. And then we can talk a little conceptually about storage. And so we can come back in, what, 15 or so?
10:15.
Yeah, 10:15, and then we'll get started close to 10. So see you guys then.
So we come back when again? I didn't hear.
We can start again at 10,
At
unless
10,
everyone's
okay.
back prior to that,
Okay.
then maybe we start early, but.
Thanks.